Hello, my name is Hyunjoo Ji, an undergraduate intern at the Steiniger Lab located in the Seoul National University, Seoul, South Korea. Today I'll be presenting on MMseqs2 profile, fast and sensitive search method to find remote homologies beyond the twilight zone. In metagenomic studies, we study uncultivable microbes by sequencing their DNA straight from environmental samples. Despite our searches against reference genomes, around 50 to 90% of the open reading frames of these metagenomes remain unannotated because sequence-sequence alignment is limited to finding homologies between sequences with high sequence identity or the daylight zone. Beyond the point where we can confidently align with just sequences or to exploit the midnight or twilight zones of homology, we need the help of profiles that represent a family of similar sequences by specifying frequencies for the 20 amino acid residues at each position. One popular method in profile alignment is HSplits, which can detect remote homology by HMM-HMM comparisons, but to scale with the exponentially growing today's database sizes, a much faster method is required. Therefore, we hereby present MMseqs2 profile, the first profile alignment method to match the sensitivity of HSplits at a much higher speed. We made this possible by extending the sim-accelerated implementation of the striped Smith-Waterman go-to alignment algorithm in MMseqs2 to support profile alignments. According to our benchmark sets, MMseqs2 profile is approximately 13,700 times faster than Hammer, while being around 23% more sensitive in a single iteration search or up to around 33% more sensitive in a multi-iteration search. It is also approximately 50 to 160 times faster than HSplits while matching its sensitivity. For methodology, I'll first explain the iterative profile search workflow and then the profile alignment alone. MMseqs2 searching is composed of three stages: a k-mer match stage, vectorized ungapped alignment stage, together called a prefilter stage, and then vectorized gapped alignment stage. In the k-mer match stage, similar k-mers from the query side is compared to the k-mers on the target side, and this step is expedited by using pre-computed index table for target k-mer lookups. If a two k-mer matches have been found on the same diagonal in the k-mer matching stage, then sequences move on to the next stage for a vectorized ungapped alignment, and then finally, vectorized gapped alignment for those with sufficiently good alignment scores. Progressively, we process less and less sequences. Now, in the iterative profile search flow, we first generate query profiles from query sequences. We've implemented a transposed PSSM search by putting the target profiles on the query side and query sequences on the target side. Profiles contain more information than sequences, and by using profiles to generate similar k-mers to be queried against a target, we improve the sensitivity. Generating k-mers from profiles can be dangerous as you might generate multiple terabytes of pre-filtering results. To avoid this, we've introduced a workflow in which we take slices of the query and then search them exhaustively without burdening the memory at once. This flow was named exhaustive search. Then after the transposed search, MMseqs2 takes the alignment results, swap its direction, and generates profiles for our original query. From then, we search query profiles against target profiles. Target profiles are represented by the consensus sequences in the pre-filtering stage, but in the gapped alignment step, we've implemented profile alignment. After the MMseqs2 profile search, we take the pairwise alignment and infer the multiple sequence alignment to build query profiles from it. We've also took after the sim-accelerated implementation of context-specific pseudo-counts and HSplits to add to our profiles for a sensitivity increase. By generating evolving query profile at each iteration, we add more diverse biological information to the profile and have achieved increased sensitivity for our iterative profile search. Next, to implement profile alignment in the gapped Smith-Waterman alignment stage of MMseqs2 search, we've extended the sim-accelerated implementation of the striped Smith-Waterman go-to algorithm. Microfrost Smith-Waterman implementation, in which SIMD registers are accessed in a stripe pattern, not just parallel to a query, helped us improve the search speed by a great amount. We've also redefined the scoring for each residue pairs of the query and target, denoted as SQITJ. We call this the symmetrized sum scoring term. Suppose you're comparing the query profile column I with the target profile column J. Then take the consensus layer of the column QI, which would be the highest scoring amino acid in that column, K in this case, and access the score of this letter in the target profile column J, 7 in this case. This will be F sub QI-TJ term, and by repeating the same for the target side, we obtain F sub TJ-QI term. By taking the average between these two terms, we have our profile scoring term for the Smith-Waterman go-to algorithm. Finally, I'd like to present to you the measured search performance of MMseqs2 profile. As for our benchmark set, we took the UniRef 100 database and annotated it with SCOP25 domains using Swipe. Among protein sequences with annotations, we extracted 500 of them with less than 20% sequence identity to only keep the ones difficult to find. To construct target MMseqs2 profiles, we've clustered the UniRef 100 database to build the non-diverse profiles, and then search this profile against the sequence database again for the diverse profiles. We've compared MMseqs2 profile against Diamond, HSBits, Hammer, PsyBlast, and BlastP. The first plot here illustrates the sensitivity speed trade-off of each tool. We took Hammer as our baseline for comparisons. The more right your point goes, the more sensitive it is, and the higher your point goes in the y-axis, the faster it is. Div indicates the diverse profiles, and it indicates the number of iterations in the legend. As clearly seen here, MMseqs2 profile searches using both diverse and non-diverse profiles outperform the rest. MMseqs2 profile matches HSBit sensitivity in the third iteration for non-diverse profiles, and in the first for diverse profiles. MMseqs2 profile is at least 50 times faster than HSBit in its highest number of iterations, and at most 160 times faster than it with a single iteration search. In addition, MMseqs2 greatly outperforms Diamond sensitivity by a huge amount, and a single iteration MMseqs2 profile search runs faster than it. The next plot illustrates the AUC curves for each tool to illustrate their search sensitivities. The more out you are to the right, the more sensitive. Then HSBits is the most sensitive tools that are being compared to MMseqs2 profile. Evidently, MMseqs2 profile searches on both diverse and non-diverse target profiles approximately match HSBit sensitivity. Profile search with diverse profiles specifically outperformed HSBit in terms of sensitivity by a substantive amount. Lastly, the third plot displays ROC1 values for MMseqs2 profile searches over 1 through 6 number of iterations using both diverse and non-diverse profiles. Starting from iteration number 3, we see a substantive boost in sensitivity, and this effect plateaus around in iteration number 5 for both diverse and non-diverse profiles. Searches were most sensitive using diverse profiles, which indicates that the diversity of the profiles may be a critical element to improving the sensitivity of our profile iterative search. I hope this makes a good explanation for some of the ongoing work for the MMseqs2 profile project. All work is available at mmseqs.com and through this QR code. Thank you for attending.